Hello, everyone. So you can hear me. We're presenting on behalf of Dave Williams. This presentation and the next one. It's from the PIPS program, Pest and Disease, managed or led by Dave Williams. My work on scab is a small component of this pest and disease sub-program. And pretty much the whole idea was that when we did the original review for this pest and disease research, it was clear that overuse of some pesticides, especially some protectants in some of the crops, not necessarily under our conditions, can have a negative effect on beneficials. And since Dave's program that the next speaker will present is about introducing beneficials, so we decided there was a need, a priority to calibrate the model that we use to predict primary inoculum for primary infections. So introducing the team here, that will be Dave Williams, and we did this modeling work in collaboration with Robert Beresford from Planet Food Research in Auckland. He's also an expert on apple scab. So what was the research trying to achieve? Well, pretty much get a more accurate prediction of primary infection risk in spring for apple and pear scab. The main diseases in fruit production really where most of the fungicides are used. And what we needed to have a reliable ascospore degree day model for use in Australia. Sorry. Improve computer-based systems to optimize spray application because you're already using some computer systems like RIM Pro and HORPlus from New Zealand. So the heart of these systems is the degree day model for predicting primary inoculum. And really, in the long term, it's better target fungicide spray programs that are compatible with IPM programs, and that includes that will be compatible with introduction of the beneficials that the next speaker will be presenting. A bit of background, degree day models capture the time and temperature dependence of ascospore maturation in the leaf litter. So I don't have time to go into the epidemiology of apple and pear scab, but most of you understand that the pathogen survives in the dead leaves from season to season. So models pretty much are used by growers for decision making to improve fungicide use and disease control in spring. We need a reliable model, the degree day model, for use in combination with other information that is needed, the rainfall, the leaf fruit growth susceptibility, fungicide residue, to help growers with identify the periods when scab is the highest risk of infection of scab, so growers can target the spray application. So stage one, pretty much I only have about 7 slides. So stage one, we pretty much did a first modeling study. We compared 6 degree day models from the United States, Norway, Italy, and models that we developed for pear scab, myself in Victoria. We published our work, and there was about 21 site-year historical ascospore data sets for both apple and pear scab from 3 countries, Australia, Belgium, Norway. This is just a summary of that study. As you can see, that what we're trying to achieve really here is that why you have the green circle here indicates the period when early, moderate, and late season varieties are developing the green tip. So we need to get an accurate model that can compensate or factor in that apple variety and the pear variety start green tip at different times. So the scab fungus is targeting pretty much susceptible young tissue. And as you can see, this red line here is the percentage, the cumulative percentage of ascospore that are produced in the dead leaves in the orchard floor. So the pathogen has synchronized its development with the abundance or the appearance of tissue. The other larger circle here is the flowering period. So when most of the tissue is sort of coming out, the pathogen is sending the spores to infect. So the model has to factor in that the different times when the different varieties are producing the bud burst and green tip, as well as the flowering period here. There's a wide range of varieties, so it's pretty much a month or longer where there's a lot of susceptible tissue to infect. So in this study, comparing these models, it was very difficult really to identify what was the best time to start the degree day model. So all these lines here represent the predictions from all the different models we evaluated here. And you can see the difference in the slopes. So, and we compare the predicted versus the observed ascospore releases. So none of the models we evaluated in stage 1 really could be used directly without intensive calibration of these models. So stage 2, what we did, we had a look at the New Zealand model. It was validated with data sets from Apple scab. So we collected the previous season, or the season before, that was 2012-13, ascospore data from 5 regions in Australia. And that's pretty much the data, the model, the logistic model using New Zealand was fitted to the data using different time to start the model. And pretty much when we fitted the model with Rob Beresford to starting from the beginning of the first ascospore trap in this study, that was the best fit we can get. But what we're really after is a start date for these models that will take into account all the early and the other late season varieties. So the best fit we can get really was a uniform fit across the 5 regions here, was to start the model on the 22nd of August. So in summary here, green tip was not a reliable indicator of the optimum biofix or the time to start accumulating degree days because of the differences in bud burst and green tip. So a uniform biofix was to start the model on the 22nd of August, and pretty much that takes into account the early season variety that's starting to bud burst and green tip. So there is a prediction of ascospores to be released when the green tip is starting to appear for the early varieties. Now we need to there are some cases of some seasons where bud break starts a bit earlier than the 22nd of August. And there was a case in New Zealand where the model really started later than the green tissue was available. So in those cases, the model has to be started at bud break instead of the 22nd. So started earlier to compensate for some varieties moving or starting the bud break earlier, which may be happening if the temperature will be increasing, as some of the speakers have been talking about. But the maturation midpoint of 50% of maturation, pretty much here when they have full bloom around here, is pretty similar to the New Zealand model, about 528 degree days. So very comparable, about 6 degrees difference. So what we found in this second study was the New Zealand model is suitable with some minor adaptation for use in Australia. And that pretty much the adaptation will be is to lower rainfall situation we have in Australia. In some cases, some regions, there will be lower rainfall. So that means that the ascospore will stay in the dead leaves and wait for the wet event to happen, to occur for the ascospore to be released. What I'm trying to say here is that this is the model, how the model looks like in an Excel format. And pretty much, again, what we're trying to do is to get the ascospore maturation and release right to identify the days with the greatest potential for scab infection, so growers can go and optimize spray application. The degree days pretty much are predicting when the ascospores are maturing inside the dead leaves, in these structures inside the dead leaves. And the pink line here, that represents the maturation inside. So the daily rainfall, we need these ascospores in there are only released into the air when there is rainfall. We plotted here rainfall in the negative axis just for illustration purposes. But ascospores that are mature inside the pseudotecium, the dead leaf, will only be released when ascospores are mature. So the analysis also indicate that we can use the same model for both apple and pear scab. The important thing is that this model can be used in Excel form, so growers can plot, use the equation and do the calculation in Excel. But the most important thing is we provided that information to Hort Plus and Rim Pro so they can calibrate the ascospore maturation model. So that's my last slide, and thanks to all the funding bodies and especially the people that contributed with the collection on ASCO Paul James, Kevin Dodds, Matthew, Peter Morrison, Alan, and Les Penrose. Thank you very much.